Hello, I'm Christian Macks. I am a postdoctoral fellow with the AI.CRED Institute. I will be presenting today on the project topic, which is fine-tuned protein language model for epitope-specific antibody design. This is a team-based project with Dr. Guidong Han, and this is the second part of the video for this project, which is covering proposed AI architecture and fine-tuning strategies. This project is mentored by Professors Chang-Ho Son and Professor Gyuri Lee from KAIST. So the goal of this project is to advance generative AI therapeutic antibody design. We'll be achieving this by fine-tuning a PLM using our in-house built clinical BCR TCR sequence database. This was curated from human formalin-fixed and paraffin-embedded archival samples. By performing this fine-tuning, we will be providing a clinically grounded antibody generation pipeline. That's our goal. In part one of this project, Dr. Guidong Han covered the FFPE sequencing and database construction. In my part, I will be covering the PLM fine-tuning strategy and the AI generative pipeline as a whole. So I'll start by giving a general intro to generative antibody design. So the primary goal of these methods is to leverage learned sequence or structure patterns to generate novel protein binders. And this is done to overcome bottlenecks that are traditionally seen in high throughput screening and accelerate therapeutic development. So in two major classes, one we have protein language models, which is the major focus of our optimization, and we have diffusion-based models. So protein language models, or PLMs, they focus on what sequences are biologically plausible to complete a masked region of a total protein sequence it's given. Whereas diffusion-based models are trained to understand what structure will fit or bind the target structure and be stably folding and interacting. So to get a bit more specific into recent advances in both method domains, we have EvolvePro, which is a PLM-based model, and we have RFdiffusion, which is a diffusion-based model. So EvolvePro follows a several-shot multi-round strategy where sequences are predicted and then experimentally validated for affinity and expressibility, and then the best candidates are passed back and iteratively improved. RFdiffusion, on the other hand, goes from a zero-shot strategy where it starts only with its target antigen and then predicts completely novel binder structures without a starting point. The shared limitation for both of these methods ultimately revolves around the fact that the candidates that are put out from the prediction require multiple expensive rounds of experimental validation in order to achieve therapeutically relevant affinities. So our pipeline, our method, is actually to use both of these strategies in tandem. So first starting with a protein, a target protein, and selecting which epitope we want to target, and then allowing RFdiffusion to generate the first candidate structure that will bind there. An antibody MPNN will translate that structure to a sequence, which then can be passed into our sequence-based mutation by an ESM-2 PLM. Those candidates can then be retrieved and then experimentally validated and passed back into the ESM-2 PLM for multi-round improvement, similar to the EvolvePro strategy. Our foreseen limitation in this application is that EvolvePro, an ESM-2 based model, is still in the context of general protein rules and not in antibody-specific rules. So we're seeking to address that limitation in the PLM's predictive capacity. So to address this foreseen limitation, we need to fine-tune the ESM-2 PLM. So what is fine-tuning? In general, protein language models are pre-trained, so they learn rules from the beginning. But fine-tuning involves giving a separate, focused dataset to update the knowledge to a specific related domain. The benefit of this is that whereas in pre-training we've learned generalizable features for all proteins and have a broad understanding, fine-tuning allows us to capture more accurately defining features of antibodies, such as relationships of CDR loop patterns to higher affinity and stability of the resulting binder structure. So specifically in our case, we have our base model, which is an ESM-2 PLM. This has captured residue level sequence features that are universal to all proteins. To begin addressing our foreseen limitation, we need to domain adapt or give immune awareness to this ESM-2 PLM, where we will give a separate database of BCR and TCR sequences that are publicly available to recalibrate for feature attention on VDJ sequences. And the second stage is to actually apply clinical context fine-tuning, where we use our human clinical library from FFPE sequencing, and this anchors the context of the model in human immune development. So performing fine-tuning can be done either just as continued training, in which you'll just give all of these sequences and update all of the parameters, but this is very computationally expensive. So instead, a common approach towards fine-tuning is parameter-efficient fine-tuning, which enables efficient adaptation of these large multi or million billion parameter PLMs to specific tasks with reduced computational resource requirements without sacrificing model performance. So I've included several main strategies seen on this slide, but the most common and efficient typically is seen as low-rank adaptation, which reduces the memory requirements while still maintaining a competitive comparison to full fine-tuning in fine feature discrimination. So to apply fine-tuning in our context, for domain adaptation, we'll use publicly available datasets that have been curated for BCR and TCR sequences. And we can achieve fine-tuning by either full parameter, continued pre-training, which is extremely computationally expensive for hundreds of millions of parameter ESM-2 model. Or the chosen method we'll actually use is low-rank adaptation, or LoRA, and this will update a small subset of highly contrasted parameter features and maintain both competitiveness and computational efficiency. In our clinical context fine-tuning, we'll use our in-house curated FFPE clinical tissues library, and we will fine-tune using LoRA. In this case, you have a frozen ESM2, and you train adapter matrices that update the attention layers for the selected parameters. And this limits overfitting on limited sample size while preserving general knowledge from the original training. And the model output is a disease context-aware model that's given a masked CDR backbone sequence and then can use the fine-tuned ESM to generate sequences with higher affinity than the generic ESM2. So this brings us to the deployment and continued development phase of this project. So in our initial deployment phase, we were envisioning designing novel binders for clinically relevant disease targets, such as cancer-related immune checkpoints, like TIGIT, or potential targeting binders, such as like non-small cell lung cancer specific targets like DLL3. In these cases, we would generate de novo CDR binder segments rather than full antibodies in the beginning and evaluate those binder segments' practical affinity by assays such as SPID and looking at their practical affinity and seeing whether our model, our fine-tuned model, performs better in shorter rounds than the native ESM2 model that has a broad protein context. The beauty of this adapted PLM is that it can be integrated into other pipelines. So in the adaptation context, the PLM can be adapted towards other biological contexts, not just TCR, BCR antibodies, but other types of small protein binders to focus on that domain. Also, a context-adapted ESM-2 PLM can be integrated into more complex AI architectures with separate models that are more fine-tuned or more focused on other aspects of the biological affinity maturation process as far as developing high affinity binders. And finally, additional functionality such as affinity ranking to further refine our final candidates can be added by adding layers that are in charge of contrastive learning between non-binders, weak binders, and strong binders. These are all potential directions that can be taken. So there's a broad potential for collaboration within the AI.CRED Institute. If anyone is interested in collaboration, you can look me up through the KAIST website. I also have a LinkedIn. If you search my name and AI.CRED, you should be able to find my LinkedIn page and contact me through there. More than welcome to discuss potential collaborations. Thank you very much for listening.